And without further ado, we'll start with our first speaker, Heidi Fettke.
All right, good morning everyone. Thanks for having me here to talk today. I'm keeping it pretty simple. It's very early on a Saturday morning and I thought we could just ease ourselves in a little bit. I like to put photos up on the top so you know who does all the work in my lab for this project. I'm part of the TPCR lab at Peter Mac, and I'm going to be talking to you today about how we can use ctDNA for biomarker research in lutetium-treated prostate cancer. All right, so we're going to jump right into it. We all know what prostate cancer is and what lutetium PSMA is, and we know from randomized clinical trials that the PSA response rate is fairly high for pre-treated mCRPC. However, only 1/3 of our patients have a durable long-term response. So why is this, and can we find a better way to stratify our patients pre-treatment? So perhaps we can identify high-risk groups, for example, for treatment intensification. Let's go next. We also know that serum PSA has little clinical relevance early on in treatment as a marker of response, nor is it always an accurate measure of disease progression in prostate cancer. So can we find, again, a better biomarker to monitor treatment response, especially early on treatment? And lastly, all mCRPC patients will inevitably develop resistance to lutetium PSMA, much like we see in all other systemic therapies. And we know very little about the mechanisms of innate and adaptive resistance as well. So again, is there a way we can use on-treatment ctDNA perhaps to investigate potential mechanisms of resistance to lutetium? And perhaps we hope that this could help us to identify novel treatment combinations with lutetium. So to start to answer these questions, we've set up a close collaboration with the multidisciplinary team at Peter Mac, so ProsTIC. They have a compassionate access registry available where eligible men with mCRPC receive lutetium PSMA. So these patients have all consented to serial liquid biopsy collection throughout their treatment. The key eligibility criteria is to have progressive PSMA positive mCRPC with a prior ARPI therapy plus minus a prior chemotherapeutic agent. So these patients on the registry receive up to 6 cycles of lutetium every 6 weeks, or 1 every 6 weeks, before they enter the clinical follow-up. So during this time, we collect up to 4 blood samples, and they go straight up to our lab in the same building. So we prepare targeted DNA libraries from these blood DNA samples, and this is so we can get a picture of the somatic landscape of these patients at all 4 time points, hopefully. So the thinking behind this, the different collection time points that we designed into the registry is what types of questions we can answer with the different samples. So for example, we hope to use pre-treatment samples, blood samples to identify markers of innate resistance, and our EOT or follow-up samples, for example, to identify or investigate mechanisms of adaptive resistance. So I have a really good pool of samples from these patients, which puts us in a very unique position to perform biomarker research. All right, so the results I'm showing to you today, they're very brief, but they're all from the registry patients. We had an initial 201 men that were screened and 150 were accepted for lutetium treatment. We collected a baseline sample from all 150 patients. From 140, we collected a pre-cycle 2 sample. And then from pre-cycle 4, we collected 76, and we managed to collect 36 on progression samples from this cohort. All right, so the median follow-up for the 150 patients was 16.4 months. We had a median PSA progression-free survival time of 6.2 months and a PSA 50 of 57%, which is about the same as what we've seen from the randomized trials. So the first thing we did with this cohort was sequence and analyze all the pre-treatment samples for the 150 patients. So this is the ctDNA landscape for all 150 patients. You can see the usual offenders here. Every column is a single patient. We've got AR as the most common, surprise, surprise. And then we took this data and we looked for any links between pre-treatment aberrations and outcomes on lutetium. So I'm showing you today 2 genomic markers that we found were independently associated with outcomes on lutetium. So patients with an androgen receptor or a P10 mutation, usually P10 copy number loss, these had inferior PSA PFS. So although no difference was observed for overall survival, I've got the PSA PFS curves up here, and you can see we've got a significant difference for both types of genomic aberrations. So P10 in particular caught our eye because of the link between the PI3 kinase pathway and PSMA expression, and there's also been some other conference abstracts that have come out since we found this data supporting the link between PI3 kinase pathway alterations and outcomes on lutetium, so we're going to keep diving into that in the near future. We then calculated ctDNA fraction for the cohort at all of the time points, which is the top left-hand side there. You can see from the graph that upon treatment, just one single cycle of lutetium after week 6, that there is quite a significant drop in ctDNA levels. So this shows us that just after this single cycle, ctDNA is sensitive enough that we can use it as an early marker of response to lutetium potentially. And then when we split ctDNA fraction via PSA response, which is the figure down the bottom, you can see there again is a significant difference in ctDNA levels between responders and non-responders for all time points except at progression. And again, this suggests its utility as an on-treatment predictor of therapy success. We also saw that high ctDNA levels at baseline had a very weak association with baseline PSA, but there was no strong correlation seen between ctDNA at baseline and any other negative prognosticators that we collected during the trial. We next stratified ctDNA fraction at baseline into detectable and undetectable. Most patients do have detectable pre-treatment ctDNA, as you can see in the red. And we observed quite a difference in both PSA, PFS and OS when we stratified the cohort this way. So patients with detectable ctDNA before they received their lutetium had a median PFS time that was approximately half that of patients with undetectable ctDNA. And we also saw a difference in these patients in PSA 50 response times, 85.2% response rate if patients had undetectable ctDNA compared to 51. So I'm pretty much done. That was very brief. I only scraped the surface of what we've been doing with this cohort at the moment. We're preparing a manuscript. We'll do a more direct comparison between ctDNA fraction and PSA dynamics, which I think is going to be really interesting, as well as generating nomograms or combinations of ctDNA variables and clinical data. Additionally, as we have the genomic landscape of patients upon the development of resistance to lutetium, we're going to be in, as I said, a very unique position to identify markers of adaptive resistance in this cohort. And lastly, the registry at Peter Mac, supported by Novartis, it doesn't end with the 150 patient analysis I showed today. Recruitment is ongoing and we're adding to it probably at the end of the year once the clinical data matures a little bit more. So finally, hopefully you guys can remember, I know it's early, but if there's anything you take away, genomic markers of poor prognosis on lutetium include AR and P10 mutations. Due to its short half-life, we saw that ctDNA levels do change rapidly in response to lutetium treatment, allowing a significant difference to be observed between PSA 50 responders and non-responders. And lastly, MCRPC patients in this cohort with detectable pre-treatment ctDNA, they had inferior PSA progression-free survival, overall survival, PSA 50, and they also had inferior PSA 90, or what we call exceptional responses. That is it for me. Thank you.